Thank you. I want to thank the organizers again for having me back here. It's always nice to come. I lived here for, I don't know, maybe 3 years or something like that. And it's always nice to visit, especially at a time of year when I can get some nice cool weather. It's wonderful. What did you say? Duke gets cold sometimes, but like here, you step out, it's crisp, it smells good. Right now, I think when I get back to Durham, it'll be like 70 degrees or something. Anyway. Okay, so this talk is it's not off topic. It's definitely on topic for the title of the conference. It is an outlier in terms of what we've been seeing most of the time. So this is it really is colloquium style. So I'm not expecting that people are familiar with persistent homology. The point is to introduce people to persistent homology and then multi-parameter persistent homology, indicate how persistent homology can and has been used, and how multi-parameter persistent homology, what we might need to do to be able to use it. That's sort of what the idea is. So these things go across the top, by the way, so you'll see them the whole time. And so I'll start by just reminding or introducing, if you've never seen it before, what persistent homology is. And I'll talk about one parameter, multiple parameters mentioned. So just one example of some statistical analysis you can do. This was old work with Steve Marron and others. And then I'll talk about sort of the mathematics of what happens in one parameter and in multiple parameters, and then what one can try to do in multiple parameters to sort of mimic what one has done in one parameter. Okay, so you're going to see this slide for a long time. It's going to bounce from this slide to lots of examples, but the input in persistent homology is a topological space that is filtered by a set of subspaces. Now I said it, so I'll give you examples very shortly, but since you have a fixed topological space, any set of subspaces is partially ordered by inclusion. That's because they're just subsets of a fixed set. Any subsets of a fixed set is partially ordered by inclusion. So whenever you have containment this way, you just say that Q is less than Q prime. So that's the Q is the set indexing this set of subspaces. And so then what you do is you take the homologies of these things. Now, just for my edification, raise your hand if you've seen homology before. Homology of a topological space. Yeah, okay, most people. But I'm still going to go over the, how one sees it and so on. If you haven't, just view this as a family of vector spaces obtained from your family of topological spaces. There's a sense in which once you've taken the homology, you can just treat it as a family of vector spaces and you don't, in some sense, particularly care where it came from. So this set of vector spaces indexed by your partially ordered set is a module over the poset. So by a module over the poset, I mean a family of vector spaces over a fixed field K. So there's going to be a field K that's going to be there the whole time. It's fixed. You can view it as the complex numbers if you want. Maybe you should view it as the field of two elements, because a lot of people do that. That means you don't have to worry about signs. And it makes it easy for the computer. And whenever you have a containment of topological spaces, or whenever you have a relation in your poset, you have a homomorphism of, or a linear map of vector spaces. And you want it to be commutative, you want it to be functorial, whatever word you like to use. The point is that if you have, if you go from Q up to Q double prime, then the linear map that you get from HQ to HQ double prime doesn't depend on which Q prime you put between them. So if you go, from Q to Q prime to Q double prime, or you went from Q to this thing up to Q double prime, the map from here to there would be the same, independent of which one you went through. That's the sense in which it is commutative. It doesn't depend on the order what you stick in between. So examples. So this is the key point. What do the examples look like? So when you have a bunch of points in Rn, then you can take ordinary persistence in a standard kind of way. And this is the way that people were thinking of persistence originally. And this is the way that a lot of you will think of when you have data sets. So this is I know this is a weird topological space to look at, but this is the topological space. Remember, we're going to filter this topological space. What is this topological space? It's actually a union of disks around these points. Yeah, so we're going to take an increasing chain of subspaces of this topological space. Here is the zeroth subspace in that chain. It is just a set of points. Yeah, let's see now, how many points, what's the homology of this thing, by the way? What's the homology of this topological space, as I've written it here? Well, it's a bunch of connected components. Anyone want to guess how many connected components? This is always fun. What? 30? Yeah, that's pretty close, actually. There you go, 31. All right, so now what you do is we're going to filter this topological space, so we're going to increase the topological space. Say it again?
He meant reduced homology.
Yeah, exactly. Yeah, but I didn't put a tilde on it. Yeah, in reduced homology, it is exactly dimension 30, he's trying to say, which is, yeah, exactly right. So I'm going to now filter the topological space, so I'm going to increase it. So what I do is I just increase the radius of the disks. Yeah, but this still, that hasn't changed the homology. The homology is still dimension 30.
You're actually in a metric space.
Well, say it again?
You're actually in a metric space because you've got disks.
Sorry, yes, so in order to make this particular, here's a general thing about topological data analysis. When people say topological data analysis, what they are doing is using topology to summarize or measure geometry. It is very rare, very, very rare, that people in topological data analysis are only doing topology. They are almost always doing geometry, but using topology as a means to get at that geometry. So it is a really important point, yes, that these disks sit in a metric space. I am constructing this family of topological spaces by using the metric to construct the family of topological spaces. But in the end, I am only recording a topological summary of each of the members of the family. Yeah, so I'm only recording the vector space of dimension 31, which is the homology of this set of disks, of the union of these disks, I should say. It's not the homology of the set of disks, it's the homology of the union of these disks. So then you increase the radius again, and the homology hasn't changed. When you increase it again, however, now the homology has changed. Yeah, so now this has only 26 connected components instead of 31. And when you're recording persistent homology, you're not just recording that, oh, here it has dimension 31 and here it has dimension 26. You want to record the linear map. So remember what the linear map is doing. The linear map is saying that, for instance, for these 2 points, there's a basis vector corresponding to this connected component and a basis vector corresponding to this connected component. Yeah, and then when you increase the radius, those 2 basis vectors have now become equal. And so the map on homology, it takes this dimension 31 vector space and maps it to a dimension 26 vector space with basis. And it takes 2 of the basis elements corresponding to these 2 disks, and it maps them to the same place in this dimension 26 vector space. And the same is true for, this pair of disks and so on. So now we increase the radius again, and then now we have a 21-dimensional vector space. So now notice that, for instance, this pair of disks, which is already only one connected component, and then this separate one, they've joined, and now all 3 of them are in the same connected component. Yeah, so it's important that we're not just remembering the evolution of the dimensions, but actually the linear maps on vector spaces that occur. Yeah, so you keep doing this, and what's going to happen is eventually you're going to get down to there's only 2 connected components now because this one disk on the lower right is disconnected, and then you increase the radius, and eventually it will become connected. Aha! But now what happened? Independent of the fact that this last disk connected to the rest of the disks. So now the dimension of H0 is 1, and it's going to remain 1, by the way, yeah? Because H0 is telling you the number of connected components. When you're taking an increasing chain of topological spaces, once you have a connected component, it can't go away. So there's always going to be at least 1 connected component sitting there, yeah? But now the dimension of the H1, so H1 is measuring loops. It's telling you how many independent loops there are, right? So where are the loops in this picture? Well, I don't know if people online can see this, but there's one nice big loop over here, yeah? Where's the other one? Can you even see it? No, there's a tiny little loop right here. Yeah, if I go backwards and then forwards, you can see that loop kind of forming. Yeah? So what's the point about persistent homology? When you look at this and you go, oh, but that hole is so small and it's there for so little time, right? Well, that's the sense. So what we say is that loop has very small persistence. It only persists for a very small range of parameters. Whereas this loop persists for a much broader range of parameters, yeah? So, but actually it doesn't just persist as one loop. So what happens when you increase the radius? So first of all, the very small persistence has now died, but this one, has now become, this loop, this big loop, has now become 2 loops. So remember the map on homology, the one loop that you had like this, mapped now to a sum of this big loop and this fairly small persistent loop over here. And when you increase the radius again, it now has gone back to just the one loop. So it goes, one basis vector gets mapped to a sum of two basis vectors, which gets mapped to one basis vector again. By the way, you'll notice that there was this other feature up here that sort of showed up just a little bit. And that's not entirely, that's not vacuous, the fact that there was a loop that showed up there. If this was a protein, then the fact that you ended up, when you increased the radius somewhat, that you got a little hole there, that maybe is an indication there's a cavity that could be a binding site for something, yeah? So the appearance of low persistence features at various scales can indicate some geometry that might be interesting, even if you think, oh, well, it was only persistent for a very little time. Okay, then you keep going and then it's just going to disappear and then that'll be it, right? Well, no, right? There's this one that showed up. It was born and then it died and there you go. So we've filtered our topological space.
Disk not at the same size.
Same rate. So if you grow the disks at different rates, yes? Yes, that's right. So we chose a particular filtration of this topological space that filtered by growing the disks at the same rate. There's no requirement that you do that. You could very well have grown the disks at different rates. And you would have gotten another filtration of, I mean, it wouldn't have filtered this exact space because at fixed radius, you'd have taken now unions of disks of various radii. But if you think of it as going on forever, then you're really filtering the entire plane. And so you would filter the entire plane in two different ways. You can filter it by, you could choose a different rate for each radius, frankly. For each of your points, you could choose a different rate. That would give you, it would give you a different persistence module. Yeah, it would give you a different answer and it would be harder to compute, but you might learn more. And we're going to do something similar, actually, where we're going to, when we do two-parameter persistence, because then there'll be multi-parameter persistence. So there are advantages to doing one parameter persistence in that the algebra is very easy. The output is easy. And I'm going to talk about what the output looks like shortly, but with a different example. Yeah, so this is a way of summarizing a data set. Yeah, so talking about what Steve was talking about when he gave his talk, this method of making a filtered topological space out of a bunch of points is a typical way of making geometry out of just a set of data points. Yeah, because if the data points were coming from a manifold and they were sampled, or near a manifold and they were sampled densely enough, then when you take the union of the balls, then you have a set that is sort of a thickening of the manifold that you started with, and then you can learn about the topology of your manifold and so on. But we are also interested in summarizing not just data sets, but data objects. Yeah, so here we're going to summarize a single data object using persistent homology. And in this case, I mean, of course, I'm drawing the parameter as being discrete, but you can view it as continuous too. It's sort of up to you whether you want to be discrete or continuous. And we're going to summarize this data object. So this data object is, I believe it's your brain. Yes, this is the arteries in Steve's brain, and it's a nice three-dimensional, yeah, the nice three-dimensional data object, and we want to summarize this data object in a way that we can then do statistics. And we want to compare this set of brain arteries to 99 other sets of brain arteries, or I think it was 97 others or something like that. But that's the idea. So we want to do summary and statistical analysis of this data object, or a family of data objects that look like this. So what we do is we're going to fill the brains, but we're not going to fill the brains by pouring blood. We're going to fill the brains by just sweeping a plane across and then coloring the points red as the plane goes up. So you start off with none of the points colored red, and now we're going to move a plane upward from bottom to top, and we're just going to color the vessels red when they're beneath that plane. So you'll see now there's no connected components, and now there are 6 connected components, yeah? 1, 2, 3, 4, 5, 6. And now you raise the plane a little bit more, and you get some more connected components. Some of the connected components have joined to each other, like this one and that one have now joined to each other. Some new ones have been born, some old ones have joined to each other, and so on. And so you get this evolving topology, and this evolving topology records something non-trivial, something very interesting about how these blood vessels are bending through space and joining with one another, if you see. Yeah. So you just keep doing this as you go up. And I didn't write down the actual dimensions of homology and so on and so forth. But I'll give you a toy example on the next slide that shows how this is going. So we're going to filter our brain by sweeping across with a plane. So there's going to be our toy model brain artery. By the way, this is the same set of points as on the previous slide, except that now I'm viewing it as a sort of a graph thing instead of a set of points. Yeah, so what I'm going to do is I'm going to sweep from right to left. And as I sweep this line from right to left, I'm going to color the vessels red as it goes across. Yeah, so here we go. So now I have, now I have no connected components. Now I have one connected component. Now two connected components, still two, three connected components. Yeah, now we're going to keep going, and now we've got three connected components still, three. Oh, now we have four connected components. Yeah, because we have 1, 2, 3, 4 connected components. And now we're going to keep going. Now, oh, wait a minute. This connected component that just started as a sort of a local minimum there joined to this older connected component. Yeah, so we have 4, but now we have 3 again. And now what's going to happen when you get to this maximum is that this connected component at the top will join to the connected component beneath it. Yeah, there we go. There we go. Now it joined. And then when you get to the very end, on the left, you're going to see that now you have just one connected component in total. Now, what are you supposed to record? You're supposed to record what's called a barcode. So here's how you get barcodes out of this. We're going to record the birth time of each new component. The birth time, I say time, but it's not actually time. Yeah, it's actually a distance that goes from right to left. It's how far the line has moved. So you're recording the birth. It really should have said birth parameter of each new component, and then you record the death parameter of each component when it joins to an older one. So here's how that goes. So now I'm just going to pretend that we don't see the arteries yet, so we're going to start sweeping. So when we start sweeping, what we do is we make a bar for each of the connected components that's born. So when we have nothing, we've got no bars. So now we start with one connected component and we put our bar, we start our bar over here. Now that component's going to lengthen, and as it lengthens, I just make the bar keep going. And now you see the bottom component has got a bar that's growing. And then, oh, now the top component has its growing bar. Yeah, so now that little tiny component has its vertex added, and then very immediately it joins to the older component. And so the bar for that tiny component has now died, right? So the component that was up here, this tiny one, it was born, and then it immediately joins to an older component. So we say it was born and then it immediately dies. So this component that corresponds to this lone dot over there persists only for a very small set of parameters. Yeah, it's born and then almost immediately it dies. And then we just keep growing these bars, and we keep growing the bars, and eventually, aha, see now when the top component has joined to the middle component, you make the bar for the top component have its left endpoint. Yeah. And so in general, then that's what happens at the end. So now at the end, what happens at the end? Now we have the entire set of arteries, and when I keep sweeping the plane to the left, the entire set of arteries is going to persist forever. Yeah? So I have to record that there's an infinite bar, and there it is on the bottom. Yeah? Okay. Yeah.
Now if you want to look on birth and death of loops, how to
Same thing. So you do the same thing. Yeah.
Okay, so
you record it
in an example a loop.
Yeah.
Decompose into 2 loops.
Yes.
So,
yeah. So the example that the example that we saw before, where you had one loop that looked like this, right, and then it proceeded to look like 2 loops that looked like this, and then proceeded to give me 1 loop that looked like this, and then it vanished. Yeah. So what you have is you've got a bar for this loop. So this loop was born at some point. Yeah. And we can draw it however we want. I mean, you want me to draw the bar going downward, or?
the thing is that when it splits into 2,
Yes.
does it mean that the top one has died? And
No, it does not. Because what happened? Because this homology class maps to the sum of these 2, and its image is still non-zero. Yeah. So this homology class, started somewhere up here, and then it persisted. But the point is, when you got the 2, now there's a second one that was born. Yeah. But then that one, when it goes down to here, this second one that was born now rejoins. Yeah. This one. So this class persists all the way from here to there.
the second one that was born, you could take a different representative, right? Okay. The other.
Yes.
The one
Yes.
left, you could take the one on the right.
That's right. Yeah. So there is a this is a confusing point to people in persistent homology. It is true that you could choose different representatives and you will get a different, you think that you get something different. However, the set of bars that you get is always the same. So this set of bars is an invariant of the isomorphism class of the persistent homology. So if you just give me the family of vector spaces, and then it automatically breaks up as a direct sum of bars that look like this. And it's always going to be this long one and this short one with a finite length there. And you can choose representatives however you want, but it's always going to come down to something that looks like that.
Why is the bottom line, this bar always growing?
Why is it always growing? Oh,
Because it's bottom line.
I was gonna ask this too.
Yeah.
It
One
seems
line
like
has
it's
to
the
grow.
second one up that you did.
this one, you're right, actually. This arrow should have been on that one. You're correct. Who said that? Yeah, you're correct. This arrow should have been on that one. I need to remember to fix that on this slide. Yeah.
You've moved from right to left, but you could be moving at any angle.
Correct.
Yeah, but presumably there's no particularly simple relationship between the results that you get at different angles.
Excellent question. So there is no particularly simple relationship between what you get from different angles. However, one of the things that people like to do is to say, well, what if I, like in the manner of a Radon transform, if I sweep across in all possible directions? Theorem, if you record the homology, so record the Euler characteristics, not even just the homology, but just the Euler characteristics in all those directions, it is what's the word for a statistic that's it's injective? It's is it identifiable? Is that it? You can recover the geometry by knowing the topology from all directions. So it is actually it is a transform in the sense of if you give me a three-dimensional surface in 3D and then you just sweep across in all directions and record the Euler characteristic in every direction, you can recover the surface from that.
So it has a well-defined inverse.
Yes.
Yeah.
Yes, that's right. Is that called what's an identifiable statistic? Is that what it's called?
No, I wouldn't use identifiability.
Okay, well, the point is it's an injection. So taking the set of surfaces to the set of Euler characteristic transforms, that's injective, and hence you can invert it. So you can, if you give the transform, then you can tell me you can recover what the surface was. So that's a great thing to do. In our particular case, in the end, for the pictures, I drew the plane as going upward, but you'll see in the statistical analysis later that actually statistics are the same no matter which direction you went. As it's roughly the same. I mean, not provably exactly the same, but sort of observed. It didn't matter whether you went from top of the brain to the bottom, left, right to left, or left to right, or diagonally. We got the same statistics either way, interestingly. Which means that really what it's saying is that we're measuring something that is rotation independent. The types of bending that we're actually getting statistics on don't depend on the orientation in three-dimensional space, which is weird because there's gravity. Yeah, but the point is that gravity hasn't affected our results.
Measure for the brain.
That was special for that particular data set and what we did particularly for that data set, yes. Yes.
But data set more than R4 is already complicated.
Yes, when you do this in R4, so the theorem I told you I think was for R3, but maybe it works for higher dimensions. I don't remember what the generality of the theorem is. Steve?
To draw off on that tangent, I wonder if there's a way you could use a graphic like this to illustrate why the persistence transform is at birth.
I mean, sorry, there is a way to do that, and people, and you know, when Cheyenne Mukherjee used to give talks on this stuff, he would have pictures that would have these, you know. So we will ask Kate Turner maybe to give a talk on that because she's on that paper as well. Yeah, exactly. So I have to remember, put that arrow on that one. Thank you. And yeah, so if you do it from, but I mean, keep in mind, we do it from different directions, you get very different answers.
Yeah.
But the point is that you're getting all the topological information from all directions. You know, the transform doesn't look anything like the surface that you started with in the same way that a Radon transform does not look like the data that you started with, but you can recover by the inverse transform. Yeah. Okay, good. So when you take these bars and then so we're going to record what the birth time of each new component and the death time of each new component when it joins to an older one. Great. And then, so this is what it looks like when you do it for the brains. Yeah. So because it's birth and then death, death always occurs after birth in, you know, usual time sequence of events. So that means that the bars always start from on the diagonal, then they go upward. Yeah, so there's a bar, there's another bar. These bars sort of overlap. They have multiplicities. Yeah, you can have two bars starting at the same time and ending at the same time. But this is what a brain looks like. This was a age 23. This particular subject patient had was 23 years old. This one was yeah. So what you get is a multi-set of vertical line segments, and they're plotted with x coordinate t, and then one for each class with birth time t and death time t prime. Yeah, so you go from t to t prime. Yeah. So this, as I say, this patient has age 23, age 32, age 49. These are just representative. I'm not drawing all 98, but just so you can see what these things kind of look like. Age 58, that's hard to see. Age 64, age 73. Yeah, so there's some trends in here that we wanted to find. That was the idea. Well, and we'll get to those trends a little bit later, but I want to talk about other kinds of persistence first.
This was just for connected components.
That was just for connected components. And there's going to be nothing else because these are arteries. And so they're not actually trees as we drew them. It turns out they're forests because it depends on how many major arteries you have coming out of the circle of Willis, which is this annular artery in the base of your skull. But you should think of them as being trees. And so since they're trees, there are no loops. However, you can fatten up in different ways. If you fatten up, if instead of sweeping across, if you fatten up, then you fill space and then you can count loops. And we actually did that too. And that turned out to be interestingly correlated with sex, as it turns out. The barcodes that we got were correlated with sex in some weird way, in a way that we don't know how to understand biologically, but it's there.
Is the algorithm to compute persistent diagram scalable to a large number of nodes?
Yes, very. Yeah, like you can do this with hundreds of thousands or even maybe millions of, you can, the algorithms these days are very good for low dimensional persistence. If you're calculating H0, the algorithms are great. For H1, the algorithms work. For H2, and for H3, probably not so much. At least not at scale. You could probably do it, but you can't do it for, hundreds of thousands of points or anything.
You need the raw coordinates of the nodes.
You do need those, but it turns out that what makes it scale, so what makes it easy to compute is the dimensionality of the space that you're computing, not the ambient space. So you could do this whole thing in a million dimensional space, but if you're only computing H0, it's still just as easy. Yeah. So the ambient dimension is not important. It's really the dimension of the space you're computing with.
What will that become more difficult if you move from H0 to H1?
Yes. Well, I mean, but it's only dependent on the 0 or the 1. If you're in a million dimensional space, H0 in two-dimensional space is just as hard as H0 in a million dimensional space because you only need the edges, the vertices and the edges, and then for H1 you only need the vertices, edges, and the two cells, and there's just only quadratically many of them, and it's not that bad. Yeah, even the dimension of the space doesn't care as long as you know what the distances are between the points. Yeah. Okay. Let's do some two-parameter persistence just for fun, shall we? So here are some fly wings. These are normal fruit fly wings, Drosophila melanogaster. They should look pretty much the same to you. These are topologically abnormal ones, and we want to do some statistical study that sort of is able to deal with the fact that these are topologically abnormal. Like the one in the middle of the bottom over here, it has a vein that doesn't quite reach the edge down there. This one on the, let's see now, the bottom one on the right has two cross veins between the second and third longitudinal veins instead of only one, like these normal wings have only one. There's one up here that has a cross vein between the first and second longitudinal veins, and actually this as it turns out, this cross vein doesn't actually reach the first longitudinal vein. So there's a question about how to label that. In any case, topologically abnormal, topologically normal. We want to do some study that sort of is able to incorporate both of these things. So here's a photographic image. This is how the data are acquired, and then what the biologists actually hand us is this. These are splines that are made from the photographic image, and they have methods of doing this automatically. So the data set that they have available has 1 million fly wings, which were. A lot of them were, so all these images were taken, by hand over a period of 20 years by various undergraduates and so on. So there's a very large data set. In any case, this is what we get. These are quadratic splines. And then, so the idea is to encode the fruit fly wing with 2-parameter persistence. Again, we're using persistent homology as a summary of a data object. So we're going to put the first parameter is going to be the distance from the vertex set. These are points in R2, and then we're just going to fatten up like we did before. Yeah, and then the second parameter is going to be the distance from the edge set. These are given as Bezier curves, and we're just going to fatten them up like that. What's the idea? The idea is the following. If I give you a human hand, and I want you to summarize the geometry of that hand, if you just sort of said, oh, well, it's basically a blob with 5 sort of protuberances, then you would lose the fact that those protuberances have knuckles in the middle. And you should remember the fact that if you take this hand and if I took this hand, but then I moved the knuckles all the way to the end of every finger, you'd say biologically that's a really different kind of gadget than having the knuckles evenly spaced on the fingers. So the idea is to remember where the knuckles are in addition to where the overall picture is. And so this is giving you summary about the biology. It's recording additional biological features by using different parameters for the vertices and the edges. That's the idea. So these are Bezier curves. Okay, so the distance is greater than or equal to minus r. This is so that the pictures of persistence come out right. What this means is that radius is going to decrease from left to right. That's what it's going to mean. But you'll see the pictures in a minute.
The important parameters. This is something that is not. I mean, you need to. It's something that you decide ahead of time. I mean, it's not.
We were talking to David Houle. So this was David Houle's data set. Ongoing is not really true. This is kind of finished because his lab is not producing data anymore. But no, the idea we discussed this with David and say what's the right way to summarize this, and we came up with this as a reasonable way to try summarizing and to try to see if we could do some biology. I can tell you about the biology after. I didn't include the slide on that here, but the biological question is really interesting. And this is a sort of a way that we were going to think of attacking it to see if we get any signal, if you see what I mean. But yeah, we can talk about the biology after. But this was definitely motivated by the biology. So the distance from the vertices, we go greater than or equal to minus r, distance less than or equal to s. As you think of r as a radius and s as a semi-thickness. So then what you do is for each choice of radius and semi-thickness, what you do is you take the subset that is near the edges but far from the vertices. So geometrically, all you do is you take the red stuff and then you remove the blue stuff. So you take the stuff that is red but not blue. So that's the topological space that you get for a fixed r and s. Yeah, so notice that if I make s, the semi-thickness, get bigger, then I get a bigger topological space. If I make the radius get smaller, or if you like, negative r gets bigger, then I get a bigger topological space. Yeah, so as the radius gets smaller, I get more red stuff. That's why I'm taking minus r here. So as minus the radius gets bigger, I get a bigger topological space. As s gets bigger, I get a bigger topological space. And so I get this summary over here, this multi-scale summary, where I have HRS, this is the homology for a fixed radius and semi-thickness. When the radius goes up, that is really minus r is going up, yeah? So when this parameter goes up, I get a bigger topological space. When the semi-thickness gets thicker, then I get a bigger topological space. So with decreasing radius, or increasing minus r, so with decreasing radius, I get a bigger topological space, and that's going to the right. And with increasing semi-thickness, I get a bigger topological space. So I have this now 2-parameter family of topological spaces, and I can take their homology. So this is a toy model of what's going on in this picture of a fly wing. So this is a toy model of what's going on. So here, what I've got is just sort of 3 veins, if you like, and 1 intersection point. So now let's think about what happens when I vary the radius and the semi-thickness. Yeah, so here's the RS plane. So when I have, let's just think about this for a minute. Well, let's do this example. So instead of having the vertex down on the bottom edge, let's put the vertex a little bit above the bottom edge. You'll notice, by the way, that you get a different looking summary. So whether the intersection point is on the edge or not on the edge makes a difference geometrically. But let's just draw this picture. When the radius is big, that means that minus r is very far to the left. Yeah? That's the picture over here. So when the disk is big, then how many connected components are there in the red stuff? There are 3 connected components, yeah? So that's the region that's drawn here in yellow. That's the region where the radius is sufficiently larger than the semi-thickness. So for these parameters RS, you get a vector space of dimension 3. Yeah. Now, when the radius and the semi-thickness are in the Goldilocks zone, where neither is too big compared to the other, then how many connected components do you see? You see two connected components, yeah, because there's the one at the bottom and there's the one at the top. Yeah, and that's in this olive-colored zone over here. That's where the radius and semi-thickness are approximately equal to one another, but neither of them is too big. Yeah, because if you make them both too big, then you either have 3 or 1. Yeah. So what happens when you have a very big semi-thickness compared to the radius? Then the veins swallow the disk. You remove the disk, but it's still only one connected component, and that's giving you a vector space of dimension 1 in this blue region up here. Any questions about that? Okay. So this is just to demonstrate that when you change the stratification, that is when you change where you put this blue disk or not put that blue disk, it changes what the summary looks like a lot. So here, if you put no blue disks, then nothing depends on the radius, and so it's all going to be just a translation invariant in those directions. If you put the blue disk down at the bottom, then you just get this little tiny olive region down here and a bit of hyperbola going up. If you put 2 blue disks, then you get the same thing, except that the hyperbola has switched its curvature. And then if you put the 1 blue disk at the top, then you get this prettiest picture. This is the one I like the best. It's the easiest to describe. Okay. Probability distributions. This is the one. So if you've stopped paying attention, now is the time to pay attention again, because this is the one where 2-parameter persistence really is going to tell you something interesting about probability distributions. So here's the idea. So if you have a given probability measure on some space M, you have some kernel of bandwidth R. See, kernels have bandwidth. This was discussed yesterday. This is a good thing. Yeah, so you can fatten up your distribution by some bandwidth. You could take various kernels that you want, but then you can convolve with this kernel, and it yields what I would call a bandwidth R expansion of your original measure. Okay, so the example is that if you take a finite sample from, let's say, finite sample from your measure, then you can approximate your original measure by taking the expansion of the sample. And so here's what that looks like. If you sample from a uniform distribution on a circle, and then you fatten it up by convolving with a Gaussian, then you get this distribution, which is kind of close to what you'd get if you took a uniform distribution on the circle and fattened it up. It just it has some variation, but it's mostly sort of, this volcano shape. It sort of goes down in the middle and so on and so forth. This is what would happen if you took a non-uniform distribution on the circle and did the same thing. Yeah, so I would ask the question, what's the difference between this distribution, with the non-uniform distribution, and the uniform distribution? What's the difference? Well, the difference is that the non-uniform one is kind of tilted in a certain way, right? But what's the difference in the support, in the geometry of the support, and the topology of the support? Yeah, you'd like to somehow say that, well, this one looks like it's kind of an annulus the whole time, and this one maybe not. So what do you do here? So what you do is you, so this is just the formula for the density. So this density, if it has a density function f, then it has support at sensitivity s. So what you do is you look at the set of points which are above some particular plane, or hyperplane if you like. And the expansion to bandwidth r and sensitivity s, what you do is you take whatever measure you started with, and then you convolve with your kernel of bandwidth r, and then you raise the plane to height s and see what the topology is of the support above that plane. So the picture looks like this. You've got these, and then you insert your plane. So here, I know it looks like it's just a circle, but it's not, depending on which height you're at. If you're at a height that's up here, then this is actually going to be an annulus, because keep in mind it goes up over the ridge and then down into the center. Yeah, so that center has a local minimum somewhere down there. So for many values of the parameter, you're getting an annulus. When you go low enough, that annulus fills in because you've gone below the local minimum. Yeah, so it's that annular feel, which is telling you that really what you're doing is fattening up a distribution that was on a circle. Really, the support was supposed to be a circle, and you can see that in this case by seeing that the homology, it's got one independent loop for many values of the parameter. Yeah, that's something that you have to do the bandwidth and the sensitivity sort of separately, because if you take your original data set and just fatten them up as points, then you're going to see sort of a bunch of spikes. So you need to fatten it up enough to get them to meet, but not so much that you fill in the inside. This is why it's useful to do persistent homology in the two parameters, that you don't have to choose, you don't have to eyeball and choose parameters. You just do it for all parameters, and then you see how persistent that cycle is. And here you'll see the same sort of phenomenon, because if this sort of low part on the right is if there's a little bit of a bump there, then for a very small set of parameters you'll see an annulus, but only for a very small set of parameters. Most of the time you'll see a sort of a crescent shape. Yeah, so this is a way of so there's because of properties of kernels, these things do actually nest as the bandwidth and the threshold, the sensitivity increase. Yeah, that's a theorem that you need. Yeah, 10 minutes. Okay. So really, okay. So persistent homology as a nice summary. It gives you this bipersistent homology of a probability distribution gives you a summary of the distribution, and the algebra and the geometry of the combinatorics of this summary tell you about the statistics of the distribution that you start from. Yeah, that's the idea. So when the poset is z to the n, this is the algebra that you get is standard commutative algebra. It's just modules over polynomial rings and these kinds of things, which I know is not the wheelhouse of people here, but I'm just saying it. When you do Rn persistence, you get modules over a real exponent polynomial ring, and that is new kinds of algebra that had to be invented just to do this kind of stuff. Okay. Statistical analysis, let's just say briefly what happens. You can reduce to linear methods when you take a 3D tree, then you get what's called a barcode, and then you get a vector in R to the 100. So these top 100 bar lengths, so if you remember those pictures, you take these top 100 bar lengths in decreasing order, then what you do is you just say write what those lengths are, and they sort of look like this kind of exponential decay. And then you take a log scale, which sort of makes it look a little bit more linear, except for the very bit from the front has some sort of noise-like big features. And then you correlate the first principal component score versus age. So I don't want to go into too much detail about this, but the conclusions are that the longest bars in older brains tend to be shorter and later. So they tend to be not as tall, and they tend to be further up and to the right in the diagram. That's more or less what we conclude. And I can show you some of these pictures, but let me show you more or less this picture. These are the PC1 scores, and you can see it's not very linear, but it's sort of linear. And the Pearson correlation is 0.52. So it's got some linear signature, but maybe not too strongly. But this is the strongest linear signature that we saw in all the methods that we tried or that were tried. I didn't. Steve tried a whole bunch of them. But it is strongly significant. The interesting part, the results are essentially unchanged when you rescale to account for variation in overall brain size, which is important because remember we're looking at heights of intervals, and the heights are actually literally their distances, and so you have to rescale the brains to make it so they're all on the same scale. You can account for known correlation of age versus total vessel length. There's a nice biological story there. And you can repeat the analysis between with residuals for regression between feature vector and total length. Anyway, all this is to say that that really what's being recorded in these persistence diagrams is genuinely topological, or if you like, topological summaries of geometric information, and that geometric information is not available in the other ways of summarizing this data, and the other standard ways of summarizing this data set. And so the moral is that you can use persistent homology to detect statistically significant geometric motifs. So the point is you're looking at all these little arcs, and the arcs are distributed sort of randomly in the brain by whatever stochastic process generated the blood vessels in the first place, and this persistent homology is recording the signature of those various bendinesses in the vessels. So what makes persistent homology work is that, every persistence module breaks up as a direct sum of intervals. Yeah, so as a consequence, you get what this barcode, Laceray persistence diagram, that's just the set of those bars that you saw. And algebraically, that's saying that your module is a direct sum of those bars. Yeah, these are old things. The first paper where I found where this comes up explicitly is in a paper on representation theory from 1980 and 1981, and there's a barcode right there. It's drawn from representation theory of the type A quiver. Okay, so in higher dimension, in more parameters, an interval is a convex connected subset. Convex means poset convex, not ordinary convex. That means that it's more or less an intersection of an upset and a downset. So this is an upset, it's closed under going up in the order. This is a downset, it's closed under going down in the order. The intersection of those two is poset convex, but it needn't be actually an interval. See, this is disconnected. This has one connected component there and one connected component there. But this one has only one connected component, and that's so this is what someone would call an interval. This we would call a lifetime instead, since it's not necessarily connected. And the statement, so a Q module has an interval decomposition if it's a direct sum of modules that look like this. So in one parameter, that's just saying that your module is a direct sum of intervals that look like this. This is an interval decomposition of a one parameter persistence module. And in multiple parameters, it would look like, if it had an interval decomposition, it would look like a direct sum of these. But you don't have to have such a thing. That's part of the problem. So let me summarize what's going to happen on this slide. It's going to say that over an arbitrary poset, every module is a direct sum of indecomposables, but the indecomposable modules don't have to be intervals anymore. In one parameter, the indecomposable modules are all intervals like this. So you take an indecomposable decomposition, you have an interval decomposition. In multiple parameters, they always have indecomposable decompositions, but those indecomposables don't have to be intervals. And what's worse is that general indecomposables are big. They're just very far from being interval modules. And what's even worse, even more worse, is that generic persistence modules in multiple parameters are essentially indecomposable plus some little tiny stuff. So then this seems to indicate that multi-parameter persistence is always giving you just indecomposable modules. What do you do with that? We want to compare with intervals. So what we do is instead of decomposing our modules as direct sums of indecomposables, which works in the one parameter case, what we're going to do now is filter our modules in terms of what you do is you lay down an interval and then you put another interval on top of it, another interval on top of it. But the way that you build the module is not by decomposing, but by filtering. So you just stack them on top of each other. I don't want to go into too much detail about this, but this is the manner in which, so this lifetime is an intersection of an upset and a downset. So this is the kind of thing that you want to stack on top of each other. Then what you do is you filter your module so that M0 is 0, M1 is going to be one of these lifetimes. M2 will be a module that has as a submodule this M1 and then something stacked on top of it. And then you take something stacked on top of that one and stacked on top of that one. So that the successive quotients, mi modulo mi minus 1, those are going to look like this picture. So you're just going to stack them up like that. And so the theorem is that you can make a stable distance out of this, or you can make a displacement out of it, where what you do is you take your modules and you can look at all possible filtrations like this, and then compare the filtrations of M to the filtrations of N, and that's going to tell you it's going to give you more or less a distance between M and N. It doesn't exactly give you a distance, it gives you something that we call a displacement. But if you take a minimum, where's the, oh yeah, that was just a stability. I was going to tell you about tameness, but I won't. The lifetime displacement is what you do is you look at a supremum. I'm sorry, you take a supremum of all the choices of lifetime filtrations of K, lifetime filtrations of M, and then you compare them, compare their distances to N, more or less. So what it's saying is that, okay, it's possible to choose really stupid filtrations of your module, yeah? Even in one parameter, you can just sort of, you can take a filtration that sort of takes, this end bit and then stacks on top of it a little bit more and a little bit more and a little bit more and a little bit more. So that the filtered pieces are just single dots in lots of places. That's not informative. But if you take an optimal filtration, then what you'll find is that this guy is a submodule, and then the quotient module of that is this guy. And you can do the same sort of thing for multi-parameter persistence modules. Even though you can't break them up as direct sums, you can filter them in such a way that you see lifetime modules in the filtration, and now you have these bars that you can compute with, a priori at least, you can compute with these bars. Okay, so what could the top 100 bar lengths mean in multi-persistence? Yeah, so the idea is that in some sense it's a sort of an obvious thing to do, yeah? So when you look for an element in one parameter, what are you doing when you're trying to find this big long bar? You're looking for an element whose persistence is maximal. You just look for a maximally persistent element, yeah? You can do the same thing in a multi-parameter persistence module. This is the last slide, by the way, so it's fine. But what's meant by maximally persistent? Well, now you're in multiple parameters, so it depends on what you mean, yeah? It could mean length, because now you have multiple parameters, so you're looking at regions in some parameter space. You could ask for the diameter of the region to be as big as possible. You could ask for the area of the region to be as big as possible. You could ask for its volume or any measure of size that you wish. And this could particularly important in scientific applications because it could be that one axis means time and the other axis means density, or the other axis means distance. And so if you have an area whose axes are, measured in seconds and, seconds and kilometers, or if you take the same area can be measured in microseconds and light years. Yeah, so like the areas, you have to take into account the scientific input as to what area actually means. So it's really important when you're doing a notion of size that you take into account what the parameters actually mean. Yeah. In any case, there are ways to then work with these things in algebraic ways, so these primary distances. These are algebraic things that you can do when you have commutative algebra power. Don't worry about the first line of this Bjerkrevik's whatever, but must an indecomposable possess a big individual element? That's the question that we're asking, yeah? If you have a big indecomposable, can you find a big element like this to mean that you have a big first bar? So what you do is you take one big bar, find an element that's maximally persistent, mod out by it, and now find an element of big persistence in what remains, and then you repeat that. And that's how you get your top 100 bars. Implementation, we have to find these maximally persistent elements algorithmically. These are algebra problems. These are really commutative algebra problems. And there's whole communities of people that work on these kinds of commutative algebra problems. This is mostly to tell you that these things exist, not to say that you should be doing it, but that we should want to be doing it so that in future meetings I can come back and say, hey, we analyzed this data set using multi-parameter persistence on, filtrations of probability distributions on whatever it might be. That's the idea. So there are implementations of two-parameter persistence. They have been used in a limited number of studies. There are now hundreds, if not thousands, of studies using ordinary persistence. And two-parameter persistence holds a lot of promise, but we have to get through some of these sort of mathematical, they're obstacles. They're not insurmountable obstacles, but we have to think deeply about how to use them, how to get around them appropriately so that we can use the theory to do something interesting. And with that, I'll give you a bunch of references. I know they're small, but you can ask for the slides and then you can read them independently later. And that's all I wanted to say. Thank you.